The biodiversity walls in Berlin, but in pretty much all natural history museums, are misrepresenting biomass a little bit. From my point of view, it would be very important to show that most of the biomass in animals is actually in these tiny little things that we tend to overlook, but that are so numerous that if you put them all together in one giant cake, are much, much bigger than all mammals and birds combined.
Have you ever wondered where we come from? And where life is going? The Museum für Naturkunde Berlin has the answers. We are reinventing ourselves for a better future. Join us. Become part of our museum evolution.
This is like a typical sample that you would get if you were to collect insects in Berlin. They may be described, they may not be described, but we are having a very hard time identifying them.
And what we want to understand is how many species are there and what are the species that are in the sample.
We take the insects that were collected and then we sort them by size. And each insect I take a photo of using this camera that will take layered pictures.
Because in the future, a lot of species identification will be done with AI models. And for that we need image data. If we don't have image data, we cannot train the AI models.
It's quite time-consuming. We've been working on a glass like this since probably about a month, I would say, if you're working on it like quite consistently.
We have thousands and hundreds of thousands to work with. So how can we solve this problem? One is, of course, robotics. Because that allows us to individualize the specimens, not by hand, but by machine.
This is the diversity scanner that can sort the Malay strap samples for now up to 3 millimeters of size. And it takes images simultaneously while it's sorting and then distributes it into our PCR plates for later genetic analysis.
We use genetic techniques, and this really involves sequencing a particular small gene fragment that can help us identify species. Processing very large number of specimens can be quite expensive. So what we have started using is called the MinION sequencer. So this is the one. The MinION sequencer is a portable sequencing device. It allows you to do real-time sequencing. The good thing about the sequencing device is that it is quite low cost to set up. What you can start doing is sequencing in your lab, and you can generate data rather quickly because the data generated can be analyzed as the data is read. So within a few hours of sequencing, you can process thousands of specimens.
And then we can use the DNA sequence to assign images to particular species. And once we have 50, 60 images for the same species but different individuals, we can start with the AI training. And in the future, we will be able to identify specimens based on images alone.
This machine is called the Compound. It's a biobanking machine. It's really useful for us because otherwise this last step of sorting all of the individual specimens into the same species group has to be done by hand. And you could ask any of the students around here that it takes really long, and it's quite a delicate process doing it all by hand because it's easy for people to make mistakes and so on when they are looking at so many tubes. But the machine doesn't make any mistakes with sorting them.
We are working with the Karlsruhe Institute for Technology on having a larger number of these robots. We are envisioning eventually having 10 robots running in parallel so that we can analyze thousands of specimens in a single day.